Okay, welcome everyone to the New England Hemophilia Association's webinar. This is part of our Webinar Wednesday series, and we're thrilled to welcome back so many of you who have attended previous episodes in this series. The whole purpose of Webinar Wednesdays is to be able to provide continuing education while we continue to experience social distancing. For those of you who I haven't had a chance to meet yet, my name is Sarah Shinkman, and I am NEHA's Program Director, and I will be the moderator for tonight's webinar. As I mentioned previously, we also have Taylor Christensen, who is joining us as NEHA's new intern this fall. She will be available to assist with the chat if people have any questions throughout tonight's session, or if you experience any technical issues. As we continue to navigate the current healthcare crisis, our primary concern is the health and wellness of the individuals and families we serve. Being connected with each other is more important now than ever. While we may continue to practice physical distancing, staying healthy means not falling into an unhealthy pattern of social isolation. Our goal is to keep you as informed as possible, which is why, as I mentioned earlier, we have continued this educational forum each week on Wednesdays, and we look forward to continuing the series throughout this fall and winter. Tonight's webinar is all about gene therapy for hemophilia. Recently, there has been an explosion of innovation in the treatment for hemophilia. It is becoming more important than ever to stay informed on all the advancements, what they are, and how they work in the body. Understanding all the options, the potential as well as limitations, risks, and unknowns of each type of therapy will enable community members to have robust conversations with their providers and to ultimately make the best decision on their treatment. We are thrilled to have two speakers from the National Hemophilia Foundation, NHF, for tonight's webinar, Maria Santaella and Brendan Hayes. For those of you who don't know Maria and Brendan, I'll give you just a quick bio and they'll share a little bit more about themselves. Maria is a research nurse at the National Hemophilia Foundation, and prior to joining NHF, she was a nurse at the HTC at the University of Miami for 15 years. And as we were talking earlier tonight, Maria brings just a wealth of information to the position. We're thrilled to have her tonight. We also have Brendan Hayes, who is the Director of Education for Innovative Therapies at NHF. Brendan is another wonderful resource. She's presented many times at NEHA in the past. And for those of you who don't know, Brendan actually was the former ED of the Texas Central Bleeding Disorders Foundation, and she has two sons with hemophilia. And Brendan is joining us from Texas, not the tropics, as I mentioned, although maybe it is our Brendan tonight. So we're really excited to have them. And NEHA would like to thank NHF for their support of tonight's presentation. NEHA is a chapter of NHF and partners on many initiatives, including our Consumer Medical Symposium earlier this summer, and events and programs throughout the year. So we appreciate their continued support and flexibility in going virtual, as is the case with most things in 2020. So before we get started with their PowerPoint, I want to just share a few reminders. Tonight's presentation will last approximately 1 hour. We are recording this session. If you would prefer not to be on camera, please turn off your camera and you will not be included then in the recording. We welcome your participation and questions. We will have attendees on mute for most of the duration of the webinar. So if you have anything that you would like to share, please feel free to add it to the chat and we will do our best to get to that during the Q&A portion. And I know many of you submitted questions in advance, and we really appreciate you doing that as part of registration. We have shared those questions with our presenters, and we will get to many of those questions as part of their presentation tonight. Anything we don't get to, we will do our best to do at the end of the session. So thank you all again for those who submitted those in advance. If you experience any tech issues, certainly not uncommon, although I think many of us have become experts in Zoom over these last 5 months. please add those to the chat. Feel free to reach out to either me or Taylor privately, and we'll do our best to help support you with those issues. You can always go on, dial in over the phone if for whatever reason you're having an issue with the visual component. So Brendan and Maria, before I turn it over to you, I'm going to go ahead and share my screen, and we'll do a couple polls. For those of you who, again, joined us and you haven't done polls with me previously, please get your cell phone or a tablet that can text, and I will pull up my screen here, and I would ask that you please respond to these questions over text. So we always start with a couple fun ones. This first one is you text the number 781-412-7971 and let us know, what are you looking forward to most during the fall? So I don't know about you all, but I've already had my first pumpkin spice latte of the season. And in fact, I was drinking it while I was coming up with this question. So that's a little hint of what I'm most looking forward to. I like the cooler temps. That's something I'm looking forward to as well. And all of the data for this is anonymous. We appreciate people engaging in this. Apple picking, that is also something I'm looking forward to. Neha Golf Tournament, which is next, or this coming Monday, the 14th. Pumpkin everything, I can relate to that. Fall colors, me too. I was previously living in DC, and now I'm back up in my home state of Maine. And so I'm really looking forward to fall actually starting at what I feel is the proper time, instead of fall beginning at like Thanksgiving as it did in DC. Awesome. Well, thank you all for responding to that. Let me move to the next question. Okay, so tell us, what is one good or bad habit that you have formed since March, since we all started staying home more regularly? So again, this will come up as a word cloud. We'll just give this a minute or so for people to respond to. And then, nice. Daily walks, yelling at my husband. I feel like we kind of on both ends of the spectrum. Watching way too much Netflix, I can relate to that. And we're also, I don't know about other people, we have continued to add on to our subscriptions. We just got BritBox, so we could watch some more British television, which I highly recommend, actually. So that's the latest in like our dozen subscriptions. Eat, I like that. Sleeping later, working in PJs, those are all things I could relate to a bit. Nice. All right, well, thank you for responding to those two. Those are our fun ones to get us kicked off. I'm going to move to the next question. So what is one word that comes to mind when you hear the phrase gene therapy, or maybe a couple words that you have, but, you know, kind of what is it that you think of? Is it, you know, a definition? Is it, you know, kind of an emotional reaction to it? That's a good one. Will it ever happen?
Sure.
These are great. And these are really helpful, especially for our presenters. It helps kind of frame, you know, coming into this session as we would if we were in a live audience, you know, what some of the thoughts are of our attendees. Hope, scary, I see, is another one. Need more info, the future of medicine. Great. Thank you all for sharing these. This is really helpful to help set a tone for as we get started here. So I will go on and move to our next question, which is, I have a strong understanding of gene therapy for hemophilia. So if you feel that you strongly agree, text 1. If you agree, text 2, 3 for neutral, 4 for disagree, and 5 for strongly disagree. And I know we'll be covering a lot of the basics for gene therapy on tonight's session, as is the title. So know that we will plan to go over a lot of information. Brendan and Maria both will share lots of different things in the spectrum of gene therapy for hemophilia. So it's interesting to see, it looks like kind of agree and disagree seem to be kind of on equal playing field here. Great. All right, thank you all. I see disagree slowly creeping up in the numbers. So it's nice to see that we've got a pretty good spectrum here of people's experience with gene therapy for hemophilia. Okay, I'm just gonna, this is our last one. What do you hope to get out of tonight's webinar on gene therapy? This will pop up as a Q&A. I know many of you submitted questions, as I said earlier, prior to the session, and thank you for doing that. And we have all of those. If there's anything else that you want to add to what it is that you hope to get out of tonight's session, that's great. And you can also feel free to add questions to the chat as we go along. So looks like so far I see, is it safe, and a better understanding. I think those are certainly ones that we will be covering tonight, some of the basics of those questions. So thank you all for submitting those. I'll give it just another minute for folks to respond to, and then I will take down my screen and we'll get started with sharing the presentation. Clarity, more info on its current standing and future, who is eligible. Right. And I should say too, Brendan has offered, if there are any questions that we're not able to get to tonight, she has offered for people to be in touch after the presentation and answer any questions that folks may have. So we'll be sure to help make some of those connections. All right, so I'm going to go ahead and stop this presentation. I'll stop sharing my screen, and we will go ahead and kick things off with Maria, who will be sharing her screen to start, and Maria and Brendan will begin the presentation. So thanks again, everyone, for joining tonight's session. Thanks again to our speakers, Brendan and Maria. It's wonderful to have you both on. Please feel free to make comments in the chat if people need anything as we go along, but I'll have you guys go ahead and get us started.
Great, thanks. We're really glad to be here, guys. So thanks so much for having us, and thanks for taking some time out tonight as well. So I know it's late there. I'm an hour behind you, but we're really happy that you're here. So while Maria works on pulling it up, I will sort of tee off the presentation for us, and just a couple of things that I'll focus on. I just want to make sure that we're all on the same page with this presentation. It is a basics presentation. However, it's a little different than what you may have seen with some of the other presentations. So, we have been seeing a lot about gene therapy really over the last couple of years. And what we're going to be doing with this presentation is expanding the conversation a little bit more. So we've heard a lot about the potential of gene therapy from a lot of the presentations that we've all been in. And, as a mom, I know I have been very interested in this technology. I've been hearing about it forever. My oldest is 24, and that's when I first started hearing about it. So it's been a long time coming. The thing that we are going to talk about tonight, though, that's a little different, again, we've heard a lot about the promise, and we're going to spend a little bit of time talking about the limitations of gene therapy as well as some of the unknowns. And we really want to make sure that at the end of the presentation that you guys have a very balanced overview of gene therapy, so that it gets you thinking, and it's going to help you when you have conversations with your providers about gene therapy as well. Maybe some of the things that we talk about, take some notes, and maybe take those questions into your provider to get a little bit have more discussion around some of those. I think that would be really healthy. And again, this is designed to provide a basic overview. We want to make sure that these complex concepts are more understandable. And then finally, throughout the presentation, we're going to have a question here in a few minutes. We're going to ask you questions. And I would love it if you guys would take yourselves off mute briefly and just shout out an answer so we all feel like we're in the room together. So next slide. So we want to develop that basic understanding of current and future treatment options. And, in addition to gene therapy, we spend a little bit of time talking about some of the other options that are in the pipeline. Because in order to make a good decision about your care, you really need to understand not only what we currently have available, but you need to understand what else is coming down the pipeline as well. So we want to make sure you have that basic understanding. We want you to be familiar with common gene therapy terms and gain more knowledge around how gene therapy works. We want you to be able to identify the potential, again, the limitations, those risks and unknowns associated with gene therapy, and then to develop an awareness of the clinical trials process, and we'll talk more about that in a minute. So we're going to start off talking about current treatment options. So here's an opportunity for you guys to unmute and shout out a question or an answer. How many different products are available to treat hemophilia A and B? Anyone have any guesses?
6.
How many was that?
6.
Anybody else?
35.
You can put it in the chat too. Thank you for the shout outs. And so let's see what the answer is. We had 20.
Wow.
So yeah, you guys are, it's amazing to me how many treatment options we have. Again, when my kids were, newborns, there were just a couple. So we've come a long way. Next slide.
That's right. Thank you, Brendan. So like Brendan said, we're just going to have this presentation will focus on gene therapy because that's what we're tasked to do tonight. But we're also going to give you a brief overview of what we have today and also other things in the pipeline. So as you probably are all familiar, we've had plasma-derived factor replacements since the 1970s. And this was a revolution all of in itself, right? It allowed for patients to treat in their home. No longer do they need blood transfusions in the hospital and the sort. So it was a huge advancement at the time. Later in the 1980s, we had we had the recombinant factor replacements, standard half-life products that were now created in the lab. We didn't have the risk of hepatitis or hep C or any viral transmissions from the plasma-derived products. And so, and eventually we got the extended half-life, right? So again, these were factors that were concentrated, they were created and concentrated in the lab. You could get 2,000 units in a little bottle, and later on they were tweaked also in the lab to make them last a little bit longer. And so that's what the extended half-life products did. And then eventually, later on, actually 2017, we had our first non-factor replacement product. And this product revolutionized or completely changed the way we thought about treating hemophilia A. And it provided a good option for those with inhibitors. So as Brendan said, we have a whole lot of factor 8 products and factor 9 products that we're currently using today. So I'm not going to go over every single one of them, but just know that we have a lot. If you have factor 8 deficiency and you have a moderate or mild, sometimes we can use thymate or DDAVP, which can be given intravenously or intramuscularly or in the nose. And then we have the non-replacement therapy, which is Hemlibra or Emicizumab, like others know. For factor 9, we have a lot less. However, factor 9 is one of those products that can last a really long time. So patients, even though they have less products, they have products that can actually last a long time. These patients are now infusing every 10 days, some of them. So it's actually come a long way from the 2 times a week. The important thing is that even though we have great products that have proven to be safe and effective, we still have more on the pipeline. All the major companies are working on ways of prolonging the half-life of these products, which we all know that not only do they work and our communities are very comfortable with them, but we also know that there's many benefits that other therapies don't have. So just so you know, we have a lot, but there's some more coming down the pipeline. So what you might not be as familiar with is that bispecific antibody or that non-factor replacement that we call Emicizumab. So there's another one like him or it in clinical trials. And what it does is it mimics the function of factor 8. So it's not factor that you're replacing because you're missing it, but it actually, when it's in the bloodstream, it actually works the exact same way that factor 8 works, right? It hooks up, it connects factor 8 and factor 10 and factor 9 together to continue the clotting cascade. So It became another great advancement. It became easier to administer it. It was less frequent dosing, smaller volumes. It provided a steady state. So instead of going peaks and troughs like we do with factor, so we give it, wait a few days, and it goes down, and then we give it again to raise up the levels. Instead of that zigzag, when you're protected at the top but not very protected at the bottom, it actually provides a steady state. So it's actually FDA approved for the prophylaxis, not for the treatment of bleed, but for preventing bleeds. And it provided a great option for those with inhibitors who really had poor ways of controlling bleeds. However, they had some limitations. It can only be given to those with hemophilia A, and again, for prophylaxis only. Factor infusions are still needed under certain circumstances. So if you have trauma, if you fall and you had your head, for example, you're gonna need factor. If you need surgeries, you might need factor as well, depending on the surgery, and so on and so forth. It also interferes with some of the lab tests that we're used to using for factor for hemophilia patients to monitor their therapy. So no longer can we measure factor VIII levels, certain forms of PTTs, which is a measure that tests how long it takes for that clotting cascade to finish the PTT. So it makes it a little bit more challenging, and it makes it challenging with people who are not,
it's gonna be water in here. What?
who are not familiar with the therapy. When you go to an emergency room and they look at your labs and they're like, oh, you look like you don't have hemophilia to me. So it becomes challenging that way. And potential side effects with other medications used to treat persons with inhibitors. And we've seen pretty severe potential side effects with those. So it's one that must be used by those who know how to use it and carefully too. So in addition to factor, in addition to bispecific antibodies, which are all, they've been approved by the FDA with the exceptions of like the ones I told you that are in clinical trials, we also have an array of new therapies that are in clinical trials as well. So we have the anti-tissue factor pathway inhibitors, which are 2 products, and the RNAi targeting antibody products. We have 1 product in that. And those products work very different than what we are used to, because it actually works with the other side of the coagulation cascade. We're not making you clot. We're putting the brakes on the clotting system. So instead of working to give you procoagulants, we're stopping the way the body stops clotting. So it's a little bit different in the way that it works. We have encapsulated cell therapy, and that is starting clinical trials very soon. And what it is they put this capsules inside the peritoneum of the person, and those capsules contain factor VIII or factor IX, and it produces the factor within the body. And what we're going to work with and talk to you about today is gene therapy. And the reason why we started talking about gene therapy in a little bit more in depth was because we, as you probably heard, we were going to get gene therapy. We were waiting for an FDA approval by the end of last month. And as you probably heard too, surprisingly, the FDA did not approve it. The FDA asked for more data from the company. And even though they had agreed that this product was going to be approved, or was going to be, not agreed, but it was going to be evaluated with 1 year of data, at the time of the review, the FDA said that it wanted more data. We wanted 2 more years of data. There was no reference as to safety issues. But as a community, of course we were surprised, and of course we were a little bit disappointed, but at the same time, it allows us to get a better understanding of what these new products are, and it gives us a little bit more information, resulting then on a product that we know and can have certainty that is going to work for, as best as it could with the best safety profile. So that's what happened last month. So we were all a little bit surprised. So gene therapy timeline, Brendan, you want to do it?
Sure. So here's another chance for a shout out, guys. So how long do you think researchers have been working on gene therapy for hemophilia?
10 years.
10 years.
20 years.
You've got 30 30 years on here.
20 plus or 30.
20.
Yeah. What's the answer, Maria?
Wow.
40 years. It was a surprise for me too, but yeah, we've been working on hemophilia for 40 years. Or I should say gene therapy for hemophilia. I know that we've really started hearing a lot about it the last few years and really, but this is technology that's been researched for 40 plus years. And, you know, don't want to really walk through this timeline, but wanted to highlight a couple things. So in 2011, that's the Factor IX trial, or the St. Jude trial, where we had our first sustained levels. And then in 2017, we had the Factor VIII trial, where that was our first success.
Right, so before we continue, we're going to give you a little overview so that we can all start from the same page. So as you all know, hemophilia is a rare genetic bleeding disorder caused by a mutation, which is a fancy word for change, in the gene that codes for the production of factor VIII or IX. And this causes a delay in clot formation. So there's a slow in the bleeding process, right? Current treatment either replaces the missing factor, so that we talk all those factor replacement therapies, we give you the factor you're missing, your coagulation goes back to normal, or imitates the way they work. And of course, as you heard me a little while ago, this only, for now, is a factor VIII. So gene therapy in hemophilia, the whole aim or the whole goal of gene therapy is to provide the body with a corrected gene. So people with hemophilia have a mutation or a gene that is not working properly. The idea is to actually give you the corrected gene and the instructions then to produce your own factor. So if you see down here, you have the mutated gene identified by the red strip. It's low or absent in hemophilia, but we give you gene therapy. We actually give you a working gene, and then you know, your body now has the instructions that it needs to produce its own factor. So that's obviously a very simplified version of it, but it's actually providing the body instructions to produce its own. So before we start explaining a little bit, going a little bit deeper as to how that works or how it's done, we have to talk about the cell. And the cell is the smallest unit of life. And everything in our bodies are made of cells, right? We have hair, we have skin cells, we have stomach cells. So we have cells all throughout. And that's why they're called building blocks of life. So each cell has a specific function. And within the cell, you have a nucleus. And the nucleus is very important because the nucleus is where the genetic materials for that cell lives. So within that nucleus, we have the chromosomes, and you probably know we have 23 pairs found that is found in the nucleus. 22 are the same in men and women. 1 is the sex chromosome, so it's what differentiates us between, you know, the sexes. And this is what's passed on from parents to offspring. And it's really what makes us what makes us us. What, you know, our chromosomes, there's the instructions that our bodies need, and that's related to everything that we are. And as you know, the gene for hemophilia is in the X chromosome, in that sex chromosome. Now, within that chromosome, you have little units that are called genes. And what these genes are, they're segments of DNA. And these are the ones where the specific instructions to everything are housed. This is the cookbook. This is the book that provides all the instructions that one needs to prepare any meal, or in our case, allows our body to function. We can also think about it in another way. You can think about it as a library, and the cell is that contains the nucleus is the library. We look at the chromosomes as to the how-to manuals. So you have the big, thick books with all the instructions in there, and then you have the DNA, which are the chapters, and the genes contain the specific instructions that makes our bodies work. So how do we get this working gene? So if our goal is to get this working gene and put it in somebody's body, how do we do that? How do we get that working gene inside the body? And this drawing represents an empty box. And the empty box, in again, another fancy name for saying empty package, is a vector, okay? So what do we know about vectors? Oh, what happened? Scientists create them from viruses, okay? They're called vectors. Once they're emptied of their viral material, they're called vectors because they're empty shells, so that they don't cause viral disease, right? The vector is used to carry then the working gene into the cell, allowing it to produce factor. In hemophilia, the most used vector is the AAV. So we like to shorten adeno-associated virus by AAV, thankfully. We have different kinds. You're gonna see, you know, AAV5, AAV8, AAV9. So there's different kinds of AAV virus. But because we use them is because they're very common. They're everywhere. They actually, most of us have been in contact with this virus, and so we've developed antibodies to it. They really don't cause disease. Once the vectors are, they remove all the genetic materials from the virus, so that vectors cannot replicate on their own. And another characteristic that it's important is that they're considered non-integrating vectors. So what it means is that it sits on the nucleus, but it doesn't become part of the genetic material of the cell. It just sits there and it provides instructions just from the outside of the chromosomes of the DNA. So we're gonna watch a little movie with Jean about gene therapy, and then we're gonna go over what that means.
Hi, I'm Jean, and I'm here to help you understand what gene therapy is. But first, we need to learn about genes. Genes are the blueprint or instruction manual of all living things and can be found in each and every one of your cells. From this blueprint, cells are able to get to work. Genes affect everything from the color and texture of your hair to your height to how your body controls bleeding. Genes are made from your DNA and carry your genetic information. Sometimes there's a change, aka a mutation, in the gene from the original copy. This mutation or defect in a gene causes it to not work properly. Genes that aren't working properly can have serious consequences. Scientists are working to find ways to solve this problem. They can replace, add, or turn on or off genes to tackle many health issues. Mutations can come from biochemical processes within the cell, something outside of the cell, or what we inherited from our parents. In hemophilia, when a mutation happens, it causes the body to not be able to produce clotting factor. Disorders like hemophilia, VWD, and other types of bleeding disorders are mostly from inherited gene mutations. When a mutation happens in hemophilia, the blood doesn't clot properly, and this is where gene therapy comes in. Gene therapy is the introduction, removal, or change in genetic material. For hemophilia, it's the introduction of a modified gene. A functional gene is made outside the body in a lab, and then it is transferred into the body. In the lab, scientists can repair a defective gene. Sometimes they add a new gene to fix a problem or even turn off a problem gene. In hemophilia, it's the introduction of a new working gene. Once the gene is modified, it changes how a protein or a group of proteins is made, meaning it changes how the cell works. In the case of hemophilia, this means the cell makes new proteins that allow the blood to clot. But to get into the cell, the new gene needs to first hitch a ride. Scientists use viruses. These viruses are called vectors, which are like little delivery trucks to get the repaired gene into your body. Scientists make a virus safe by removing all the stuff that can make you sick. Then, the delivery truck, aka vector, can carry the repaired gene into the cell, and your body can make the clotting factor that helps your blood to clot. Gene therapy is bringing us closer to a reality where we have new options to treat rare disorders like hemophilia.
Well, I hope you like Jean. So let's talk a little bit about what Jean just went through. over. So this model, again, it offers a very oversimplified version of gene therapy, but hopefully it illustrates the point a little bit clearer for you. So the first step is the development of a working gene, right? So we're in our cases, we're interested in hemophilia A and hemophilia B, so factor A and the gene for factor A and factor 9 production. So we create that gene in the lab. And then we create a vector or a package, right? And this vector is very important because this vector has to enter the body and deliver the gene where we want it to deliver without really causing any trouble. So the working gene is placed inside the vector and it's literally infused in the patient. It's a one-time infusion done in at this time in an HTC that is part of the clinical trials. So the body enters, the vector enters the body, and remember how I told you they'll go to the cells and sit right next to inside the nucleus, and it starts producing factor 8 or factor 9. Well, the target cells are the livers. So these vectors are created in a way that they actually know to get to the liver. They want to get to the liver, but it's given into the patient's bloodstream, which means that it goes everywhere too, right? It goes into the blood, and it finally, hopefully, reaches the liver. So the new gene instructs the cell how to produce the new protein, which is very exciting, right? Because maybe for the first time, that patient has never ever made factor 8 or factor 9 in its life, and it's now producing factor 8 or 9 on its own. The issue with this is that it also, I'm missing one. It also results in the production of factor, but it also, the body has to get rid of this of the vector, right? So the vector is delivered to the liver. It reaches the liver cells like it's supposed to, like we want it to, and then the person starts getting rid of the vector, and we get rid of the vector via our saliva, our urine, our stools, and this sometimes you're going to hear that as referred to as vector shedding. Okay, so you're shedding the vector, the empty vector now. So here's the whole system. So why is hemophilia suitable for gene therapy? Well, it's because the defect is caused by a single gene mutation, right? So one gene. We know there are many mutations that cause hemophilia, but all those mutations exist in one gene. So we know exactly what to target. Factor 8 and factor 9 are very easy to monitor, just the way you get monitored when you go to your HTC. They check a factor 8 or factor 9 level. And we also know that small increases in factor levels can improve quality of life. And we know this because our severe patients, when you compare severe patients with our moderate or even the mild, their quality of life is drastically better. So we have the potentials of gene therapy, right? So this is the ultimate goal, to provide the body with the instructions on how to make your own factor, hopefully to normal levels. We eliminate spontaneous bleedings and reduce other types of bleeding, right? So, and we do that, it also reduces or eliminates factor infusions. So we eliminate spontaneous bleeding and perhaps minor bleedings, and reduces the need for factor infusions. And the reason why we do that is what we talked about before. It provides gene therapy. Your body is making its own factor, so it provides that steady state. No longer are we doing peaks and troughs. Not necessarily you need any factor infusions. That's the goal, and it actually provides a steady state. And ultimately, it improves quality of life. Imagine a world in which you don't need to infuse yourself a few times a week or every other, whatever the case may be. So it provides peace of mind and the ability to participate in many activities that one otherwise would not. However, there are limitations, and there are risks, and there are unknowns, okay? So there's a variable response, even within individuals who are getting the exact same product. We don't know yet what that's the case. So there's variability in between products because they're a little bit different, but there's also variability within the same product within people, okay? There's pre-existing immunity against the vector antibodies. Remember how I told you that they're very common and many of us have been exposed to that virus before? That means that many of us have antibodies. So antibodies is our immune system recognizing that virus and saying, uh-oh, I don't like this virus. It might potentially cause harm, and it attacks the virus. And so if you have antibodies against the factor, against a vector, then gene therapy will not work for you. The inflammatory response or the immune system response. Well, this is related to, we think, to the high doses of gene therapy that are needed to actually raise levels to a level that is likely to cause no bleeding and reduce the infusions. So doses are as high as hundreds of trillions of genome vectors per kilogram. So if you imagine that an adult weighs 60 kilograms in average, that means that you're getting hundreds of trillions times 60. So it's a huge dose, and the immune system goes, can potentially have a big reaction to it. It also causes, at least at this time, we have limitations in redosing, because we know that when we give you that vector, even if you didn't have antibodies before, you're going to recognize that vector and you're going to develop antibodies. So that means that at this time, we don't think that you can be redosed, at least not with the same product. So that might change in the future, but for now, that's it. And remember we talked about vector shedding? Well, if you are shedding vector after gene therapy, you could potentially infect your siblings that may want to have gene therapy later on, or anybody in your family, your spouses, and so on and so forth. So patients that undergo gene therapy also are asked to use barrier methods of contraception, because one of the ways that we can, the vector is being found is being in semen. So that's why we ask them to wear to do protection before when they're having relationships. The vector can impact non-targeted cells and could show up in other cells in the body. So what does this mean? This means that even though we've created these vectors to go target the liver and to go directly to the liver, because we're infusing them in the bloodstream, they can end up anywhere else, right? And although this is not the intent, it's certainly something that can happen, and we don't know what the repercussions of that would be. Also, genes could potentially incorporate or be inserted into the host DNA. Remember I said that one of the great things about AAV vectors were that they were non-integrating, so they did not integrate into the genetic material of the cells. Well, we know from other trials that there's some insertion that occurs in the cells. Again, we haven't seen anything serious result from that, but it certainly can happen. May potentially be harmful to the liver. So again, remember I told you about all these big high doses. So then they're all being targeted to the liver. So in addition to that huge immune response that many patients have, and that has been controlled until now with steroids use, high dose steroids, or sometimes for a prolonged period of time, it can actually, we know that the liver enzymes go, raise when after gene therapy. So they recover also with steroids, but we need to be, mindful of that, or what that may be, the need to study that a little bit more. And we have unknown long-term side effects. So even though we've been working on gene therapy for a long, long time, human trials have only, been around for like 10 years. And so, there's a lot that we potentially don't know. We don't know the cost of it. We have an idea. The company that was about to launch in August gave us an idea, but we really don't know what that's going to cost and how insurance companies will pay for that. And then I think that the biggest lesson here is that it's once given, it cannot be undone. We in the hemophilia world are used to getting factor and perhaps switching factors if you don't like one, because we know that after hours, your body gets rid of that factor, right? In gene therapy, once you get all these millions of vectors, they're part of you forever. That gene that gets into your cells, it's part of you forever. So it's important to understand when you're going through clinical trials that all these are things that can happen. Of course, we haven't seen any serious side effects from any of these therapies, but they can. Some patients have been have needed the steroids for the immune responses, etc., and their liver enzymes go up. So, Brendan, you want to take on the gene therapy? Who's eligible?
Absolutely.
Okay.
So, we've talked a lot about, you've learned more about gene therapy, and a question that we get asked at the National Hemophilia Foundation by payers is, how many people are going to want to do gene therapy? How many people are going to be eligible even to do gene therapy? So we decided to create some information in this presentation just to sort of help you understand that. So next, please. So if we start off with the population of hemophilia that we've all heard, that 20,000 figure, which by the way, based on some research that WFH has done, that figure is more likely to be between 30 and 33,000 people in the United States that have hemophilia. So if we start out with that number, next please. So first of all, we know that at least for the product that is farthest along, it was for hemophilia A, although we do have products for hemophilia B closely behind. So then we think about the inhibitor population. And the data that I'm sharing with you now, this is really based on what we've learned from clinical trials. And typically, the FDA approval will follow along with that, meaning that it's going to be closely aligned with what happened in clinical trials. So with clinical trials for gene therapy, there's no inhibitor patients enrolled. And even if you've had an inhibitor that's been tolerized, you're not eligible for gene therapy, again, in the clinical trials as they exist. There is a clinical trial that was looking at inhibitor patients, but I don't believe it's enrolling anymore.
No.
Next,
No,
please.
it hasn't started to enroll.
Oh, it hasn't started, okay.
Mhm.
Then we have to think about comorbidities. So, with clinical trials, we're really looking for that really healthy person, right? So, you can't have diabetes, for example, or heart disease. So those are just a couple of examples. In regards to HIV status, HIV status doesn't prevent participation in some of the trials if it's under good control. And again, the same thing with hepatitis C. If there's been a lot of damage to the liver, you probably won't be a candidate for gene therapy as it stands now. Next, please. So also, it's for the adult male population. So this isn't indicated for children or a pediatric cohort as, again, as we know it today. There certainly is talk about what would gene therapy look like for a pediatric population, but again, it's adult males, no females. So that's another limiting factor. Next, please. And so, Maria spent some time talking about, that pre-existing immunity to the vector. So developing those antibodies. So if you have the antibodies, you won't be eligible. And again, these are all things that you will sit down with a provider and talk about and get that additional testing to check all of this to find out if you're a candidate. So in reality, we're really talking about a much smaller number than that 30,000 figure. So it's probably going to be less than 10% of the population that has hemophilia will actually be eligible to do gene therapy.
And Brendan, just to interject a little bit, because there was a few questions about trials that were pre-submitted. So even though the company that was supposed to get an approval, we all thought that they were at the end of August, they have continued their clinical trials. They're gathering those 2 years of data. So it doesn't mean that people have stopped working on gene therapy. It just means that more data is going to need to be presented to the FDA for them to make a determination. So even, all the companies are still working on this. Has COVID impacted this? Absolutely. So nobody that has been enrolled in the gene therapy was left out in the cold, but I believe that they're not enrolling new patients, still not to this day. So even though clinical trials are continuing and data is still being gathered, COVID has impacted them in that way. And then the other question was that what happens if you participated, so let's say you participated in a clinical trial with a product with gene therapy and that gene therapy just didn't work for you, it didn't go well, can you redose? Can you use another product? And at this time, that's not part of the inclusion criteria. So you have to not have a period of a year, I think it is on most trials, with no other investigational agents. There's still that question then, since all of gene therapies in hemophilia, they're all AAVs or some form of AAV, having you receive one product that is, let's say, AAV5, can you receive an AAV7 or 8 or whatever? We don't know that, and because you might have antibodies against that product, and again, that antibody makes it mute. So, so I think those are the only questions that I have here for. If not, feel free to ask at the end later.
Great, thank you, Maria.
So is hemophilia a cure is gene therapy a cure for hemophilia? So obviously that is the big question. What do you think? Yes, no, I'm not sure. Feel free to put it in the chat or shout it out.
C.
Anybody else? Don't be shy.
We've got a no in the chat.
No or
Couple
no.
no's.
Okay. So yeah, let's find out, right? That is the big question. But at this point in time, it's definitely not a cure. We're calling it a durable treatment. And so, like I alluded to before, the resulting factor levels are not the same for everyone, even those who are treated with the same product. Factor levels may only reach the mild to moderate range. So we have patients who respond really well and are in the normal range, where some patients that don't respond very well and are in the small digits. And we have patients who initially respond really well, and throughout the lapse of time and months, those levels decrease. So at this time, we're trying to figure out what that all means and how we can predict better which patients will be doing better with those products. But unless we know that, it's really very unpredictable. Besides, we still need factor for injuries and surgeries. If your levels are not within the normal range, you're still gonna need factor, perhaps not every day, perhaps not in prophylaxis, but you will need some. And like I said, it's not known how long this therapy will last. And most importantly, gene therapy can be still passed on to your offspring. Remember, we're not integrating anything into your genome. We're not making, we're not fixing or editing your genes. We're actually just giving you instructions to be able to produce your factor 8 or factor 9, but we're not doing anything that will prevent you from passing the hemophilia gene onward. So a quick clinical trials overview, since all of these therapies are undergoing clinical trials.
Okay, so, what is a clinical trial? So it's a research study conducted with volunteer participants to evaluate any new medical treatment, a medication, or a device. And if you think back to the beginning of the presentation when Maria was going over all of the innovation that is happening now, not only gene therapy, but all these other products that are in development. It's really important that we understand the clinical trials process, because you will likely be asked to participate in clinical trials, and it's just good to know a little bit more about them. So we're just doing a super high level overview. NHF does have a more in-depth presentation on clinical trials, but for the purpose of this presentation, we just have a couple slides. So really here want to showcase that there are steps that you have to go through, and these things take a long time. It can take 8 years or more to get a drug through this process. So one of the things that it's really important for us to remember is that we have responsibilities as participants as well. And, especially when you're talking about some of these innovative therapies like gene therapy, it's really important that you understand the informed consent process. And now more than ever, it's important that you really read all of the fine print and ask questions and understand that, you'll need to report all of your health changes through this process. You'll need to make sure that you keep all of your scheduled appointments, and especially in the beginning, these are pretty intense. And so just make sure you understand the requirements of participating in a gene therapy clinical trial. Next slide. So talking a little bit about participating in a hemophilia gene therapy trial, one of the things that you will do is you'll have conversation, to find out if gene therapy is a good option for you. And so this is a time for you to really unpack all these questions and all this information that you've been gathering about gene therapy, and find out, talk to your provider and find out if it's something that would be good for you. Again, making sure that you obtain that informed consent process and that you meet the criteria. The other thing to think about too is these clinical trials aren't going on at every HTC across the country. So it's really important too that you understand that travel might be involved. And again, your HTC can help you find a trial near you. Additionally, you can go to clinicaltrials.gov. It's a little cumbersome, but there's a wealth of information in there about all the clinical trials and the studies that we've talked about tonight. So, there's a time limit too in screening. So you may find out that you're not, that you don't have the antibody. So, but you do have a short window between the time that you get your testing and when you have to make that decision and get the infusion. So again, just understanding all of that. And you will be closely monitored the day of, as well as long-term follow-up. And, it's really important to make sure that you understand that this is long-term follow-up. This isn't just something that you'll do for a year. You'll need to be followed for many years to come, because you'll want to ensure that you know about any changes or any adverse events that happen with these trials. Next slide. Okay, so we're winding down. We're coming to the end. So let's find out if you guys learned anything. So hopefully you'll get all the answers right. So anyone can take part in a gene therapy clinical trial. True or false? What do you
False.
think?
Somebody's paying attention.
Absolutely.
That's right.
The answer is false. So, current trials, we talked about it a little bit. They're limited to males 18 and older. Females are currently not eligible. It's severe to moderate patients, mainly severes, no history of an inhibitor, minimum number of exposure days to factor that you have to meet. They want, healthy patients, and you may not have participated in another gene therapy clinical trial. So I'm going to let Maria do the next one.
In gene therapy, a vector is used to transport the genetic material. True or false?
True.
True.
All right. So again, the answer is true, right? A vector is used as a package to deliver the working gene, the instructions on how to make factor. Study participants who have had previous exposure to viruses. So if you've been in contact with viruses, again, and they're everywhere. They're outside, they're in the park, they're in everywhere we live. And you may not be eligible if you have antibodies against the factor, against the vector. So we're trying to research this. We're trying to understand it a little bit better. And it sounds like there's people that have very good ideas as to how we can redose. But at this time, that is not possible. But yes, vectors are how we get gene therapy into the body.
False.
After receiving gene therapy, are you cured for hemophilia?
False.
False, that's correct. So gene therapy is not a cure. Every person's response to gene therapy might be different, including no change in factor levels or a minimal increase in factor levels, right? So I think that this is important to know when if you're interested in getting clinical trials, and thankfully people are interested in getting gene therapy, because without them, this science would never progress. But I think that it's very important that you enter the trial knowing that it might not necessarily work for you. Okay, that your levels might not be what you want. Obviously, everybody wants to be in the normal range, but that's not always the case. So it's very important to know this from the get-go. And we don't know how long the effects, even if you have a great response, we really don't know yet how long they will last. Bren, you want to do this?
Sure. So once I receive gene therapy, I never have to take factor products again. Is that true or false?
False.
Diane, you've been designated the question answerer for the group.
Okay.
Great job. So yes, it's false. So, you know, as we've learned tonight, responses to gene therapy are very individual. If adequate levels are reached, factor may not be needed for potential bleeding episodes. You know, we've heard some pretty amazing stories about people who've had gene therapy in clinical trials. But if those normal factors aren't reached, factor replacement therapy may still be needed. So, you know, if you have a potential surgery or you have some sort of an accident or injury, you may still need factor. And, you know, at this point, it's unknown really how long the factor levels will be maintained with gene therapy. And, you know, we're going to continue to study and learn more about how long gene therapy will last.
So the most common side effect of gene therapy includes fever, elevated liver enzymes, development of antibodies to the vector, immune response to the vector used, or all of the above.
E.
Okay, anybody else? Thanks, Diane. You've been, so yeah, so actually, this is kind of like a trick question because I did not talk about fever. I always forget to mention that the immune system response is usually comes accompanied with fever. So that's definitely a side effect. Liver enzymes, I already told you that we're targeting the liver at really, really high doses. So the liver enzymes do go up in most patients, again, treated with steroids. And the idea with it is to make sure that, that we can control the response. Development of antibodies to the vector use, very common. Immune response to the vector use, another common thing that happens. The immune system recognizes these viral vectors and says, oh my God, what is this? And attacks it. So the answer is all of the above. But good guess. So Brendan, want to take over?
Yeah, so finishing out, you know, one of the things that I didn't hit upon that I just wanted to briefly touch on is that, you know, another question that we're hearing a lot from payers is, you know, we talked a little bit about the potential of gene therapy. So they're trying to figure out if it's worth their investment. So, you know, having a little bit more data, having a little bit longer could be a good thing. So, we wanted to share some of the gene therapy resources with you guys. I put the link in the chat. So we've got frequently asked questions. We have, you know, we have got a glossary. We've got that gene therapy video. We've got a great webinar that goes into a lot more detail about gene therapy from Dr. Pipe. We try to keep news articles posted, and there's all kinds of stuff out there. We also are going to be putting some of the presentations from our bleeding disorders conference. So if you missed those, we had a couple of really great ones as well. So next slide. So, you know, in summary, we hope you enjoyed the presentation. I know it seemed a little bit sort of like a downer, but we really want to make sure, you know, this presentation is designed to make you think. And to ask more questions. So you have time now to really dig in and ask more questions. And so hopefully we've given you a lot to think about. So advancements in treatment options are going to continue for the foreseeable future, which is so exciting for our community. So even though we had a little setback with the FDA, we're still all moving forward and we're all excited about the potential. So, you know, again, seek to learn more about existing and future treatment options. Talk to your provider, make sure you're able, you know, you've got all your questions so that when you go in for your next appointment, you can learn more as well. And, you know, try to stay on top of all the clinical trials that are underway. So Maria opened our eyes to quite a few that are happening, and be a good idea to keep on top of those as well. And then again, we want to make sure you understand that long-term commitment that's required for gene therapy. So this isn't just a one kind of, you go in and get gene therapy and you don't ever have to go to the HTC again. You'll need to be followed and you know, you'll need to be tested to make sure that your factor levels are where they need to be and all of that good stuff. So if you want to learn more about risk and benefits of these current and future options, please talk to your provider. They're the best folks who can give you the right info for you. And, again, please reach out, look at our website for more information. And I want to make sure that we thank all of our sponsors that have generously, provided funding for us to do this presentation tonight. So again, thank you guys so much for being here. And we've got a couple of minutes if you have some questions, if Sarah wants to do that. We tried to cover as many as we could. We got quite a few questions at the beginning before the session tonight, and we tried to answer as many of those as we could. So hopefully we did that.
Thank you.
Thank you. Thank you.
Thank you.
And thank you, Maria and Brendan. That was wonderful. I feel like I attend a lot of different sessions on hearing gene therapy, and every time I do, I learn something new. And just hearing the information over and over again. I mean, I think, one of the biggest things, takeaways for me, is that understanding gene therapy is like learning a whole new language. There's so much of this terminology and concepts that are complicated and ever changing. And so, I think just maintaining, this communication and education is so important, so that, as you both referenced, going in to have conversations with your providers, you can understand some of the basics of what they're saying. It's much like the advocacy work that we do. We would hate for people to not feel like they at least knew the basic terminology going into having these critical conversations. So thank you both very much for that. You did answer, I think, pretty much all of the questions that were submitted in advance. So I really appreciate that. There was one question that came up to me as you were speaking. Neha is actually doing an event in Spanish next weekend, our Hispanic Heritage Symposium, and this being Hispanic Heritage Month. I wondered, does NHF have resources available to folks who speak Spanish that we might be able to share with that with our community in the future? Is that something that you could access through the website?
We actually don't have resources translated into Spanish yet, but that will be a focus of 2021.
Great. Okay. Excellent. I will make people aware of that so they know something to look forward to in the future.
I was actually recruited to be Jean in the video, so I'm still thinking about it.
Oh, well, I have no doubt, Maria, you would do an excellent
Maria
job.
does Jean in Spanish.
Yeah. I'm sure you would be a great resource for that. So that's good to know. Wonderful. And then, Brendan, I know you kind of referenced this a little bit, but insurance is always a really big topic that people, have questions about. I know someone had asked it as a pre-submitted question, but just, I think you mentioned on your website, if people have questions about how do we pay for gene therapy or want to submit questions, is there a place that people could go to specific to insurance for gene therapy as a follow-up?
We're actually, we have a payer relations team that's working closely with payers to, help them understand more about gene therapy, help them understand more about that eligible population, and we're doing everything that we can to educate payers and help them understand how important gene therapy is to our community. Just the thing that we all have to remember is we do have great products, but they don't work for everyone. They're not perfect. And, we still don't have a tremendous quality of life like people who don't have hemophilia do, right? So it's important that we continue to move forward with innovation. And part of that is ensuring that we find ways to finance. And so we're very involved with MIT. They have a New Digs consortium. It's called a Focus Project. It's a think tank to try to work with payers, not only private payers, but public as well, so your Medicaid, Medicare. So we're doing all that we can to work closely with payers, and our hope is that this will be a therapy that will be covered in the future.
Great, that's great. And I know earlier in this session you mentioned the World Federation of Hemophilia. You just referenced that acronym. Have they been doing some work on gene therapy as well? Is that another resource for people who are interested in what's happening on an international scale to check out?
Yes, I don't have the website. I should make sure I do that. But yes, that's another excellent resource.
Great.
WFH, absolutely.
Awesome. Well, well, I know we're getting after 8 now, and I want to be mindful of people's time, especially as many of us continue to spend a lot of time on virtual learning these days. So with that, I'll close out the presentation and just once again thank Maria and Brendan for their time tonight and all of the information that you. If you have additional questions and want to get in touch with Brendan, please reach out to Nia and we can help facilitate those conversations. I wanted to just put a quick plug in for next week's session that we have planned, which will be on making connections and coping mechanisms during social distancing as we all continue to stay home. So I'm going to put into the chat the registration for that. We are looking forward to that session next Wednesday, same time, same place, Zoom, 7 p.m. So the recording of this will be available on our YouTube page, usually within 24 hours. We get that up on our website. Really appreciate everyone being here tonight. And it's also nice to hear Brendan and Maria ending on an optimistic note of some of the things to come. I think especially in this time of COVID and with recent news, it's nice to hear that there is some optimism that we can all kind of think about moving forward. So I hope everyone enjoys their evening and best wishes as many of us start the school year with kids and other shifting priorities at home. Let NEHA continue to be a resource for you. We're continuing to update our COVID page on our website, and we are happy to provide any assistance that you and your family may need during this time. So hope everyone has a great night, and thanks again.
Thanks, everyone.
Bye,
Bye-bye.
everybody. Thank you.